Here I am in Tinbergen Studios giving you the lectures on learning. There are two of them. First one's on the basics of learning, and the second one's on more complicated things. So this class will be a brief overview of learning, roughly the topic of how the brain uses experience to shape relevant behavior. We will look at both associative and non-associative learning. In associative learning, an animal links together two kinds of events. So linking two events together. In non-associative learning, there is no such linking. We will look at habituation, which is the waning of response to repeated presentations of the same kind of event. We will look at sensitization. That's the opposite. It's a form of non-associative learning in which the responding increases. So we will look at how a danger signal increases an animal's responding to subsequent unrelated stimuli. Then we'll look at two forms of non-associative learning, classical conditioning and operant conditioning. In classical conditioning, an animal links events in the world. So two different events in the world are linked with one another. One is used to predict another. In operant conditioning, that's a matter of linking one's own behavior to outcomes. It's learning what to do, doing something to get some outcome. We'll start with habituation. So ongoing hum, there's a bit of hum in this room that might be recorded on the video. Hopefully you'll soon pay no attention to it and you'll be paying attention to my words. Right? That illustrates habituation at work. Happens a lot in real life. In this particular study, the scientists collected urine from pigs. So it's a kind of a messy experiment in which they have to get urine samples from a lot of different pigs, and they're not taking the piss out of science either. This is a real experiment with some purpose. So what they do is then present samples of subject pigs, right? Samples of urine to these subject pigs for a minute, and they measure how much the pigs sniffed the urine, right? So what you see is that the sniffing level went down from trial 1 to trial 2. Okay? When you repeat the presentation of the same urine from the same subject, the pigs take less interest in it. They habituate. On trial 3, if you continue to give them the urine from the same pig, then habituation continues. They show little interest in sniffing. But if you give them a different pig, then boom, they show more interest once again. So among other things, what this experiment shows is that pigs can tell one another apart. They can discriminate other pigs on the basis of the smell of their urine. We will look at sensitization in this sea slug. It's called aplysia, made famous by this man, Eric Kandel, who won the Nobel Prize for this work. Kandel himself hails from Austria. He's of Austrian heritage, but he escaped to the USA, escaping the Nazis. And he spent a long and productive career at Columbia University, which is in Manhattan, New York, and is one of the Ivy League universities on the East Coast of the USA, so one of the best. He had nothing but scathing comments about his home country, Austria, in their treatment, in their refusal to stand up to the Nazis. Okay, so at any rate, Austria's loss was America's gain because his discoveries won him a Nobel Prize. This is a great system, this aplysia, for understanding a neurophysiology of learning, but we will only look at the barest bit of neurophysiological detail in this class. So here we see the top side of aplysia, so you're looking down at the animal. They got these flaps called parapodia. Parapodium is the singular. Right here, the flaps, the parapodia, are pulled back. The head here is at the top end, in case you can't tell the head and the tail of this animal. Tactile stimulation of these structures that are associated with the gill, these things, will lead to a gill withdrawal reflex. So this is pulling the gill out of the way of possible danger. This happens in the real life of these animals because they're swimming around these coastal waters and there's a lot of debris like seaweed, and they sometimes get in the way. So this kind of reflex does happen in the real life of the animal. All the work I'm telling you, however, is done in the lab. On this graph, we see both habituation and sensitization in this system. But we will only look at briefly at the neurophysiology underlying short-term sensitization. On the left there, the top line is showing the extent of gill movements, right, on repeated trials. So what you see is habituation happening over repeated presentations, say repeated stimulation of, say, the siphon, right? Same stimulation over and over again, the gill withdrawal response gets smaller and smaller. On the right there, so something happened between trials 13 and 14, and that's giving the animal a tail shock. So zap. The animal gets zapped, and you see that on trial 14, the shock has sensitized the gill withdrawal reflex. And this can last from minutes, a few minutes to a few hours, depending on the parameters of the shock strength and those things. Okay? Note again that the tail shock here does not cause the gill withdrawal. It causes an increase in the next time you stimulate the siphon. Okay? So the change is on trial 14 when you stimulate the siphon, then the gill withdrawal increases. So in this system, we have one interneuron here from the tail and one sensory neuron, say from the siphon, the experiment we've been, the example I've been using. And there's one motor neuron here, this big thing, which actually commands the gill to withdraw. So this thing orders a gill withdrawal response. In fact, there's quite a lot more than this, but for our purposes, it's good enough to pretend that there's one of each kind. First thing to note is this thing on the bottom right here. The motor neuron in this system responds in a graded fashion. What that means is that if you depolarize the motor neuron, you don't have to actually get it to fire, to undergo action potential. The more you depolarize it, the more it triggers the gill withdrawal response. So the amount of gill withdrawal response is basically dependent on how much you depolarize this motor neuron. The interneuron from the tail synapses onto the axon of the sensory neuron, as is illustrated here. So let's say that the sensory neuron, as our example, relays information from the siphon, right? What the interneuron does is releases a neurotransmitter, the biogenic amine serotonin, and serotonin then does something to the sensory neuron. There are a lot of other circuits involved in this system, but let's focus on this one to give you the barest bit of neurophysiology. So we'll ignore everything else and focus on this system. So what happens at the sensory neuron? You already know that neurotransmitters can partake in direct chemical transmission or in indirect chemical transmission. Of course, in this system is likely to be indirect, because that's the route that makes all kinds of interesting things happen. Direct transmission just opens some ion channels, and that just would not have the desired outcomes. Okay? What serotonin does is bind on the membrane of the sensory neuron, and it causes then a signal cascade. We'll skip the details of the signal cascade. I want to jump down to this point that's illustrated here. So what it does is make the potassium gates don't work as well. That's what impedes potassium gates means. Right? So the potassium doesn't move through as easily now as before. Okay? I remind you that down here there are voltage-sensitive calcium channels that let calcium ions into the neuron. And calcium ions are implicated in neurotransmitter release, something we already learned in week 5. So you might take a minute here to deduce what consequences this has the next time an action potential comes down the sensory neuron. Well, our starting point is that potassium gates don't work as well. The neuron is then slower to repolarize. Okay? And that means the action potential basically lasts longer. And that has the consequences that the calcium channels then stay open for longer. And if the calcium channels stay open for longer, then more calcium enters the neuron. And if more calcium enters the neuron, then here's the bottom line. You get more neurotransmitter release from the sensory neuron. And, all right? You get more neurotransmitter release from the sensory neuron, and hence this would depolarize the motor neuron more, and you get a bigger gill withdrawal response. So the bottom line is serotonin causes the sensory neuron to release more neurotransmitters, right, the next time it gets stimulated. So that's the change that underlies short-term sensitization. We'll now move on to associative learning and start with classical conditioning. This form of conditioning is often called Pavlovian conditioning as well. The two terms mean the same thing. So Pavlovian conditioning is, of course, named after its discoverer, Ivan Pavlov of Russia. Pavlov was a Nobel Prize winner, but I think I already told you he didn't win his Nobel Prize for this discovery, for discovering classical conditioning. He won it for some work in digestion. When he came to discover classical conditioning, he had already got his Nobel Prize. You could say he got the Nobel Prize for the wrong thing, because classical conditioning is certainly much more famous. On this topic, it's mostly a matter of learning these key terms with a few key learning phenomena that I'll throw in. What we start with is a reflex-like linkage between an unconditioned stimulus and an unconditioned response. Okay, that's what the animal comes in the world wired with. Right? In Pavlov's study systems, hungry dogs will salivate when a bit of food is put in their mouth. All right? The CS, the conditioned stimulus, is usually some kind of neutral stimulus, like a ticking metronome. And what the animal does is learn to connect this CS with the UCS. So CS is used to predict the coming of the UCS through this training process. And then that generates a conditioned response, behavior that the CS comes to elicit from the animal after classical conditioning has taken place. So at the first presentation of the conditioned stimulus, such as the metronome ticking away, that doesn't elicit salivation in the system, but it does do something. It elicits an orienting response, sort of a "what's that" response. The dog sort of looks around, sniffs, and wonders what's going on with this metronome going on. Repeated presentation of that will lead to, yes, you've got it, it will lead to habituation. So this orienting response will disappear after you keep presenting the metronome by itself. But if you follow the metronome predictably and very shortly after the metronome onset with some biologically significant event, like a bit of meat powder introduced into the mouth, that is, with some UCS, the animal then comes to predict this biologically significant event on the basis, right, of the CS. Eventually, the conditioned stimulus alone will elicit some preparatory behavior for the predicted coming of the UCS. In Pavlov's case, the dog salivates, right, to the metronome before meat powder arrives, exhibiting what's called a conditioned response. And that is the process of classical conditioning. And originally, Neutral stimulus comes to elicit a response that it did not previously elicit. Let's now turn to a few cases to give these terms some meaning for you and to let you learn about the basic phenomena of classical conditioning. On the left there is a honeybee foraging naturally, and on the right there's a honeybee harnessed in the way it is for classical conditioning. It's receiving a drop of sugar water there. This conditioning paradigm works best if the conditioned stimulus is an odor or a mechanical stimulus, right, applied to the antenna. So the antenna are these bits sticking out. Okay. The antenna has both mechanical sensory receptors and chemical receptors. So for our example, let's use an odor, a classic conditioned stimulus used in this particular paradigm. In the training, what you do is you waft the odor across the antenna. Say, let's use peppermint, waft the odor of peppermint across for a couple of seconds. At the end of that period, you then offer some sugar water to the antenna. Okay, so there's a bit of sugar water being offered. And once the bee senses the sugar water with its antenna, it then sticks out this long tongue-like thing, this proboscis, right, to suck up the sugar water. That's why it's called a proboscis extension reflex. So the bee sticking out its proboscis to suck up sugar water. Okay. So you train the bee a few times this way, right? Odor, sugar water, odor, sugar water, and then the bee learns to stick out its proboscis in anticipation of the coming of sugar water. Here are some data on the acquisition process on the left here. In fact, acquisition is very fast. Each of those C's there is one conditioning trial. And basically after two pairings of the CS with the UCS, with any of the chemicals, so in this experiment, sort of pure chemicals were used as CS rather than peppermint or complicated things like that. After two of these pairings, the bees have basically got it. Some 80% of the bees anyway stick out their proboscis in anticipation of the odor before the sugar water arrives. Okay, that's shown here. There's a test here shown in gray, and that's presenting the odor, the CS, without the presentation of the sugar water, the UCS. This just confirms what was already evident on trial 3, that the animals are sticking out their proboscis in anticipation of the reward. So not all bees learn. You see it's only 80% successful. There's a small proportion, it seems, that just don't get it in this artificial setup. On the right here, we see the phenomenon of generalization. What generalization means is that the bee exhibits its conditioned response not only to the conditioned stimulus that it was trained with, but also to related and similar odors as well. Just take, for example, the training to hexanol. So that's this dotted line here. The bee sticks out its tongue not only to hexanol here, but also to heptanol a lot. Okay? Heptanol and hexanol, fairly similar compounds. So, so that shows the phenomenon of generalization. They are not very clear. They are not very excited, however, about nonanol. This bit here, they don't react very much to nonanol. So that's showing the opposite, what's called discrimination. Okay? Now, why generalization? What I want to argue here is that this is not a mistake in the system. It's actually a design feature, and it's just about found in all learning. So all learning just about shows this kind of generalization. Look, the bee in this situation is trying to predict what the world is like. It's trying to predict important things like whether a stimulus is going to deliver sugar water. It's not doing a discrimination test, right? So don't think of the bee as doing a discrimination test. It is trying to predict what class of stimuli will predict the coming of sugar water. And basically, it's betting the heptanol is similar enough that the bee is betting that it should predict sugar water as well. Okay, so that's the way to think about it. That's the function of generalization. Try to classify the stimuli that will deliver the goods It's not. It doesn't serve the bee to do discrimination studies. It helps the bees trying to predict the world. Okay. Extinction is another key phenomenon in classical conditioning. So in the extinction procedure, after you've trained up the bee, the animal is then presented with a CS, right? The odor in this case, but which is not followed by the UCS in this case. So presenting the CS alone is the extinction procedure. The result, as you can see, is the diminished probability of showing the CR, and that's shown in this block of CS-only trials. Over trials, the proportion of bees responding decreases. So, does that mean the learning has been wiped out? No, not at all. The phenomenon of spontaneous recovery, shown on the right here, shows otherwise. So look at the black bars here. These were the bees that were retested in this conditioning paradigm. So this is without any further training. So the experimenter just waited some time after these 5 extinction trials and then presented the CS alone again. And you can see that after some time has passed, say 2 hours, 4 hours, some proportion of the bees have recovered their conditioned response. Okay? So in this case, the bee hasn't forgotten, right? This just shows that the bee hasn't forgotten the training that it's had. Again, you could think of it as the bee, looking back on its past experience, if you like, to anthropomorphize a bit, and trying to bet whether this new stimulus, after all this delay, is going to predict the coming of sugar water. Right? It's got competing hypotheses. The very last bit of experience I've had, the last 5 trials says, no, it's not going to come, but the history before that shows that it is going to come, right? The 3 acquisition trials before that. So the bees under some conflict, right, of evidence after all this delay. So that's the way to think about spontaneous recovery. We mentioned methylation last week. Methylation here actually plays a role in the retention of classical conditioning in this particular paradigm in honeybees. This has been recently discovered. The basic idea here is to block methylation and look at retention after 24 hours after training on this paradigm. So methylation usually requires a particular enzyme, DNA methyltransferase. You don't have to know the name. Right? So when it's got an ASE ending, that's generally some kind of enzyme. So this enzyme works for transferring methyl group onto the DNA, right? Pretty much as the name implies. So you can then zap the bee with a drug that blocks the action of this particular enzyme. That's called zebularine, Zeb there. And then you could compare that with applying an inert solution to the bee, basically a solution with no drug. Okay. So what you find is that it doesn't matter when you apply this particular drug. You can apply it before the training, during the acquisition, after the acquisition, just before you do the testing. Zeb, the enzyme blocker, has a small but significant effect when you take it over all these treatment times. So methylation seems to play some role in retention of the memory for classical conditioning. Okay, this is only found at a retention test after 24 hours after training. The same drug actually has no effect on acquisition at all. It only affects the memory process. So you could also see that it doesn't wipe out the memory. Okay, so there must be something else that's preserving the memory without methylation in the case of when a drug is applied. Okay, so at least it shows a role for methylation in learning these data. We'll look at one more. Oh yes, I should have clicked that, but but that just summarizes the data. You get a small effect of the drug on the 24-hour retention test. We'll look at then one more conditioning paradigm. Fear conditioning in rats and a little bit in humans as well. So the UCS, when you use it for a rat, is shock. So you see all these bars here? What you can do is run electricity through it, and that scares the devil out of the rats. It hurts a little bit, but it's not really the hurt that's scaring the rat. It's somehow this shock is translated into a danger signal. The rat thinks there's something going after it and fears for its life. So that's why shock works so well across a whole range of animals. You can then pair a neutral stimulus, for instance, a tone generated from a speaker or lights flashing in this Skinner box, and pair that with a shock, right? Present the shock a few seconds after the tone, let's say. And as you might suspect, the conditioned stimulus comes to elicit fear, and does that very quickly. Fear learning is very fast. So fear is a central state, and it has many behavioral consequences, right? Only some of which I will list. So in the autonomic nervous system, well, you should already know which branch will kick into gear when animals in fear. Yes, the sympathetic branch. In humans, you can also measure what's called galvanic skin responses. This is basically your skin sweating when you're under a bit of fear. So with the right instruments, you can measure sweaty palms. That's what that does. Startle response. Okay, I forgot to bring my gun, but if I shoot off a gun very loud here, you will startle. So our startle response sort of the whole head, whole body movement. You can't help it. You'll scrunch your shoulder even when you know it's coming. Okay, so that's the startle response. A rat that has a startle response, so rat literally jumps, right, like that. And what fear does, if the rat is in the fear state, then the startle is potentiated. What that means is it gets bigger. So a fearful rat jumps higher with noise or any kind of sudden stimulus than a non-fearful rat. And you can see, of course, that's adaptive, right? So if a cat is jumping on a rat, then boy, a potentiated startle might be the difference between being in the cat's claws and being able to run away and show fear, potentiated startle another day. Urination and defecation, right? We say a really scary thing scares the shit out of you. Well, it's actually got physiological reality, especially in rats anyway. So rats do a lot of this. This last thing you want to think about is bowel control when you're about to be chased, bound up by a cat. If you want to, you can actually measure the level of fear in a rat by counting the number of pieces of feces in the operant box after conditioned stimulus is given. Psychologists aren't very fond of counting shit, so they often use this freezing response as a measure. This is again an instinctive rat response. So what it means is actually the rat stays stock still when it's in fear, and you could see what function it has. What it's trying to do is stay motionless and be as inconspicuous as possible. It's trying not to hit the motion detectors and the visual systems of predators, if you like. Okay, so if it's freezing, then it suppresses ongoing behavior. So if you have the rat doing something like pressing away for a reward, you can measure the amount of behavior suppression as a measure of fear, and that's used by psychologists a lot as well. Oh, let me tell you one experiment on human fear conditioning. Well, it's sort of an experiment. The experiment was actually on something else, and this is actually from Louis Barker, who wrote a book on learning. So here he is. There's a photo of him strapped up. This was in days when he volunteered to do some of this, be a guinea pig basically for the U.S. defense. And the experiment wasn't on classical conditioning, it was on the crashworthiness of these vehicles. So what happens, he's strapped in there and the siren goes off for 30 seconds and then the thing goes zoom sliding down a rail and crashes into a wall, right? And he's the test subject in there. Barker reports that he gets fearful responses anytime anything that sounds like that siren goes off. So there you have a case of human fear conditioning. All right, We now turn to operant conditioning, the last of our topics for this class. It is sometimes also called instrumental conditioning. As I've already said, Edward Thorndike was credited with starting the business, and Thorndike had cats solving puzzle boxes, as shown on the left here. But B.F. Skinner really pushed the course here in publishing a lot of data and in developing automated boxes that can record the behavior of animals, often now called Skinner boxes. So this automated recording instrument led to a blooming of research, and there's now just piles and piles of data in operant conditioning on all kinds of animals, typical, especially pigeons and rats, the psychologists' favorite lab animals. So if you want to mine the data, there's lots out there to test theories on. We have three key terms here to learn in operant conditioning. Let's start with the bottom two here. In general, the animal is doing something to get something, and it learns about the relation between its behavior and its consequences, what it gets. What it does is called the operant, which is that which is necessary to get the consequence. And it's called R for response, the response that the animal has to make. What it gets is called the outcome. So in the one letter symbol is O. It can be a good or a bad outcome, which we will talk about on the next slide. So O for outcome. A good outcome is a reinforcement, and a bad outcome is a punishment. And then we have the S plus, the discriminative stimulus. So this rule of doing something to get something usually only applies in certain situations, right? And that's the cues of that situation. Well, that's a discriminative stimulus. So in an operant box, you can make food a rewarded outcome, for instance, and bar pressing for a rat as the operant it has to do. So, and you could also make it the rule that this relation between bar pressing and getting the food only works when the tone is on. The tone then is the discriminative stimulus. It tells you that in this situation, when the tone is on, then this contingency of reinforcement is enforced. Then bar pressing will get a reward. If the tone is not on, bar pressing will not get a response. Right? So we do this all the time. So when we discriminate situation in which behavior will or will not get rewarded. So talking when you're in a pub with friends, that's fine. You get reinforced for that. Talking in the middle of a lecture, that's not so good. Although hasn't stopped some of you from doing that. All right. So that's the discriminative stimulus. It's like a green light signal, right? It tells you when it's okay to do a particular behavior. Okay, then we come to contingency, reinforcement, and punishment. This is a lot of technical jargon, and some of it's very confusing, but unfortunately, you've got to learn it. Those are the terms we're stuck with from learning theory. So there's a 2x2 table here, each box with a term in it. So a reinforcement in this jargon means there's something good. A punishment means something bad. All right, so that's pretty intuitive. The other differentiation, positive and negative, is much more confusing. So positive here means there's something that's added, and negative means there's something that's taken away. So positive and negative here have nothing to do with good and bad. All right, so positive reinforcement is sort of the ideal that Skinner has in mind, sort of an ideal for clinical practice, if you like. You like to get your client working away for something that he or she desires. Let's start with some example. Let's start with negative punishment. Okay, so negative punishment means taking away something that the subject likes. So that the event is taking something away, and the event is not liked by the subject. Okay, so parents use this kind of punishment when they tell kids, "No ice cream tonight. You've been too naughty." All right, or they could put them on the naughty spot for a few minutes. That's basically a timeout, right? A timeout in which they don't get to interact with others, they don't get to play. Timeout is a classic form of negative punishment, and you can instill that in an operand box as well. So basically, you turn off the rules of the game. You tell the pigeon you can't peck a key for reward for a few minutes, or you tell the rat you can't bar press for food for a few seconds. Usually a few seconds rather than a few minutes. Okay. Positive punishment. Ah, yes. So positive punishment is a punishment that's an aversive stimulus. So it's presenting some stimulus that is aversive to the animal. This aversive stimulus is contingent on some behaviors. This is generally used to reduce the frequency of that behavior. Okay. A word from our sponsor now. This lecture is brought to you by Stop, the ultimate yap stopper, based on the power of positive punishment. Do you have a flatmate who just won't shut up? Is the student next to you drunk again and rattling on endlessly? Now you can combat endless yapping with Stop, the ultimate yap stopper based on the power of positive punishment. Just fill your Stop with its ready-to-use paralytic agent and collar the culprit. Every time the blimey yapper makes a sound, Stop detects the vibrations and sends out a shot of paralyzing agent. Your yapper's voice box is put out of commission for a few minutes. Use a stronger dose for a more permanent solution. Stop, guaranteed effective because it is based on the laws of positive punishment, available from the Central Intelligence Agency. Conditions apply. Well, in fact, it's an anti-barking device that you use on dogs, and what it squirts out is a bit of citronella, and that's enough of a punishment for dogs that they can reduce the barking. All right, so the device is strapped around the dog's like a collar, and it's sensitive to vibrations. So when the dog barks, the vibration triggers citronella, a squirt of citronella that works to stop dogs barking. Jolly simple, a great device. Wish my neighbors knew about this. Negative punishment. Now this is negative reinforcement. Let's get this right. Negative reinforcement is usually a confusing term. But negative reinforcement is actually a good outcome. It's a good outcome because something unwanted has been gotten rid of or has been prevented. So it's basically, there's two forms of it. There's escape. So escape is working to get rid of something that's unwanted. And there's avoidance. Avoidance is working a way to prevent some punisher from occurring. Okay. Now, a typical device for studying avoidance and escape is this kind of a shuttle box. So here's an example with dogs, for dogs on the right, and there's an example for rats on the left. So what you do is you get the dog to predict when shark's coming. Is say you sound a speaker, you send the tone on for 20 seconds, and then the floor gets shocked. The dog gets shocked, runs around frantically, eventually jumps over this hurdle, right? And the dog escapes. The other side is safe, all right? Then the speaker goes on again for another trial, and another 20 seconds later, the dog then runs around and again gets very frightened, jumps over the other side. That's why it's called a shuttle box. It gets to jump back and forth to try to escape the shark. Okay? Eventually, the dog actually wises up, and when the tone goes on before the 20 seconds is up, it jumps over. So the dog then learns to avoid. It's very interesting behavior, right? The speaker goes on, dog jumps over. There's no reward or punishment of any kind, but this behavior can persist for a long time. So explaining this has caused some consternation among psychologists. Avoidance is actually a very powerful motivator if you want to use it in self-control. That's this line on the bottom here. The most powerful form of this kind of avoidance contingency runs as follows. You write out a check for a small amount, say $5, to an organization you intensely dislike. I don't know, you can support Colonel Gaddafi or the Ku Klux Klan or the generals in Burma, some people you don't like. Okay, so you write out this check, you sign it, you put it in a sealed envelope, and you give it to an enforcer, one of your friends, right? And then the rule is, well, you got to do what you're going to do, right? You're going to do your 3 hours of studying this week, or else it gets dropped in the mailbox, okay? This kind of motivator, this kind of negative reinforcement motivator is very powerful for self-control. So if you want to use it, well, be my guest. Finally, positive reinforcement is doing something to get something that's wanted. That's positive reinforcement. Positive reinforcements can be primary or secondary, okay? So a primary reinforcer is intrinsically rewarding. It's food to a hungry animal. It's water to a thirsty animal. It's sex to an animal that's in heat. A secondary reinforcer, on the other hand, is rewarding by way of association with the primary reinforcer. So animal trainers generally use this form of secondary reinforcer. So the first thing you want to do if you want to train an animal is use something like a clicker or some noisemaker that you can tell the animal quickly it's done the right thing. Right? So what you do is you pair the clicker with a bit of food first, right? Do a bit of classical conditioning like this. Then the clicker takes on positive properties. It takes on rewarding properties, and you can use it in training. So that's used to train animals to do all kinds of tricks. So you don't want to be feeding the animal on every time it does something right. Sometimes you just want to tell it with a clicker, right? And just follow it occasionally with food. That's how animal training is done. And it's also generally done by shaping, this last point here, shaping by successive approximations. So what you do if you get one of these killer ways to do this is first you got to reinforce just approximations there. Even if the animal just swims toward this end, you sound the clicker and you give it a bit of fish, right? Eventually you basically raise the bar and expect them to approximate this behavior even more and more. You then wait till they actually touch this particular landing spot before you give them a reward. Eventually you wait till they actually slide on. And then you can add this bit about tail raising and so on. So slowly you raise the bar and require more and more precise behavior in order to get them rewards. So that's how you can train an animal to do these fancy things like you see in these various shows. There's an urban myth about a group of students who actually try to train their lecturer this way. So, the lecturer will be talking away and sort of moving to the left and right. And basically, every time the lecturer slide it to the left, and they would start, making noises and scratching their ears. And every time he moved to the right, then, they would play rapt attention. And eventually, the lecturer is all the way over this side of the room, and not realizing why. Try that if you dare on one of your classes. Okay, a few more key phenomena. Generalization. So again, so this now refers to the discriminative stimulus that's used in training, right? So in these particular experiments, the pigeons were trained with a particular color of a key, all right? And they get to peck at that color to get a reward. So let's say, let's take an example. I think it's 600 nanometers here, although it doesn't look very clear on this slide. Probably looks good on your PDF though, right? So that's what the pigeons have learned. They learned to peck at this discriminative stimulus. What you can ask after you train them this is that after you go through this training is, well, will they then generalize to other discriminative stimuli? And the answer is, well, yes, if they're close enough. Okay, so they're close, you get a good amount of behavior, of key pecking out of the pigeons. But pigeons are actually quite sensitive here. So if it's, if it's a few tens of nanometers of wavelength away, then their response really drops in this case. Okay, that shows discrimination. So responding to similar stimuli as the discriminative stimulus, that's generalization. Not responding to quite different stimuli, that's discrimination. And again, this is not a mistake in the system. Again, just like in the case of classical conditioning, the pigeon's trying to pick out, classify the world, trying to predict what will deliver the response, what will deliver the reward, right, for key pecking. So it's basically generalization is the bird betting that, well, it looks similar enough, I bet picking this will give me some reward as well. Okay? Once again, we get extinction. So if you stop giving the bird, in this example, the reward altogether, then eventually its later responding will disappear. Okay, it will get lower and lower. But be warned for any of those parents who want to try it in terms of extinction on bad behavior in their kids. This might take quite a while, and things can get worse before they get better. Right? At first, when the bird doesn't get reward, it actually tries harder. It pecks more and more and more and thinks, oh, maybe it just haven't pecked hard enough. Okay, so often in extinction, the behavior can persist for a while before it gives up. But eventually, the bird will give up. But does that mean the bird has again forgotten what the training? No, you get spontaneous recovery again. So if you train the birds on, say, 20 sessions of this key pecking, and the 21st session you now put in extinction. Now the bird can peck all it likes and it doesn't get any reward. Right? Over the course of an hour session, the bird will give up responding. All right? Take the bird back home, put the bird in the box next day again, turn on the key light, and what will happen? Yes, spontaneous recovery. The bird will start pecking again. So this again, like classical conditioning, the bird is betting, well now, yesterday it wasn't so good, but the 20 days before that I got rewarded, so bugger it, I'm going to give it a go. All right, so that's what we think is happening with spontaneous recovery. The bird's looking over its whole body of experience and deciding that, well, yes, it's worth a try again. Okay, let me summarize this class then. We looked at non-associative learning and associative learning in this class. Habituation is a form of non-associative learning. It's the diminution of responding with repeated stimulus presentation. The response rate goes down. Right? Other kinds of events sensitize. We looked at tail shock in increasing or sensitizing the gill withdrawal reflex in aplesia. And the mechanism here is called presynaptic facilitation. Tail shock causes a change in the presynaptic neuron, right? The sensory neuron we've called it, such that it releases more transmitter the next time the action potential, next time it undergoes action potential. Well, next time it gets stimulated, right? And releases more transmitter, and that causes an increased response in the motor neuron, which causes an increased gill withdrawal reflex. We looked at classical conditioning. This is the matter of predicting what events lead to what other events in the world. Animal learns that a CS, a conditioned stimulus, heralds some important event, the UCS, or the unconditioned stimulus, and it exhibits a conditioned response. In operant conditioning, an animal learns that doing something produces some outcome in some situation. And that situation, right, is characterized by the discriminative stimulus, the S+. Other key terms here are R for the response and O for the outcome. We looked at 4 different kinds of reinforcement contingencies, and please review that and get them right on the exam.